And so it's our great privilege to welcome our new director of the Harvard University Herbaria, Dr. Jeannine Cavender-Bares this evening, and Bob Wernerell, our vice president, is going to introduce her. Thanks, everyone.
Thank you, Melissa. Yeah, just a little background on our speaker tonight and our guest tonight. She had her undergrad degree at Cornell and then a master's degree at Yale in forestry and came to Harvard right here. So she's coming back to old stomping grounds and did her PhD at Harvard here in 7-year period with Professor Bazaz, who's a well-known plant ecologist. She was in grad school here with Elizabeth Farnsworth, who was very instrumental and key to New England Botanical Society and also Go Botany and the Flora Novae Anglii, all the illustrations. And also another grad student that was here at the same time was Tristan Seidler, who's at UMass Amherst Herbarium. So she was with some good people that are connected to us. Did a postdoc at the Smithsonian Environmental Research Center, which is a great place, and then further at the French National Center for Scientific Research in Montpellier, which we're going to hear more about, I hope, because I asked her to talk about that tonight. They do a lot of great ecological papers coming out of that institute. Then she went to University of Minnesota, which is one of the big Midwestern land grant institutions that are fantastic for research. They have lots of place to grow things and good soil. So you can do a lot of research there and a lot of forests as well. She has over 200 peer-reviewed journal articles. And then she was there for 21 or 22 years, still some connections there, I think. So, and then she came with a new hiring from the Salata Institute here that focuses here at Harvard on climate change in an interdisciplinary approach with engineers and some other, I think some humanities as well. So that was a new hiring of 8 new professors. And now she'll be following in the footsteps of previous herbarium directors such as Asa Gray, Charles Sprague Sargent, George Lincoln Goodall, William Gilson Farlow. The Farlow Herbarium is named after Oaks Ames, and Charles Davis, of course. It's a big job. It's one of the largest herbaria in the world, a collection of various herbariums here with over 5 million specimens. And of course, a big chunk of those is under Lisa Stanley's direction for the New England Botanical Society collection. So with that, I would like to welcome Professor Cavender-Bares.
Thank you for that introduction, Bob, and thank you for the invitation to be here. And I see that the title I gave you a long time ago was a vision, and I will give you a vision, but I'm mostly going to tell stories about where I came from and who I am and get to the vision at the end. Hopefully it will all make sense by the time I get there. And with that, I'll just get started. And here's kind of the outline. So I'll tell you a bit about me. This is what Bob asked me to tell you, like, where did I come from and how did I get here? So I'm going to do that. I'll tell you about past and ongoing research, which might help explain why I wanted to come here. And I'll explain a bit about the Biodiversity and Planetary Stewardship Initiative and the Harvard University Herbaria in the context of that. And then at the end, I'll get to the goals for the Harvard University Herbaria. If I start rambling on, I'm going to just skip ahead if I start doing too much storytelling. So, all right. So where did I come from? Well, that little dot in southeastern Ohio, small college town, Athens, Ohio, where Ohio University is. That's where I'm from. So I grew up in a small college town in Appalachia. Atul Gawande, have you ever heard of him? Maya Lin, Joe Burrow with the Bengals. Those are the greats from my hometown. It's a pretty diverse place. We lived outside. it's really lovely weather for much of the year. And there are a lot of oak species there, as it turns out. I don't even remember the first time I met an oak tree, but that's a picture of my two sisters and I sitting in an oak tree in the middle there. That little kid, my little baby sister, is now the director of the Huntington Botanical Garden in Pasadena. Somehow the botanical vibe really passed down in our family. I drew this, I don't know, when I was a kid. So I was somehow thinking about oak trees and their interactions with mycorrhizae and fungi so as a kid. So that was all, I guess, being a daughter of a mycologist, you're thinking about fungi early on. My family camped everywhere. We were always outside. I always heard about other kids who got to stay in hotels, but we were always camping. And that little kid in the middle is me. One of the reasons we were always camping is, and I didn't realize this until years later, but my dad was always collecting soil samples. And it was just a lot easier to do the soil sample collecting if you're camping than staying in a hotel. So there was a lot of that. And then there's my grandpa, my physicist grandpa, trained at UW-Madison, where he met my grandma, who also did a master's degree at UW-Madison. And well, just go on to my grandpa. So that's John Cavender. And he got a job at Bell Lab in New York City in 1930, and he was just ecstatic. Then the Great Depression hit, and then that was bad times. And then he ended up working in an engineering for Western Electric during the war years. And then at some point he like got fed up with a rat race and quit and bought a farm in upstate New York on the Vermont border and like had a few cows, and they barely made it. It was like subsistence farming, but he read a lot of books and lived to be 97. He died right about the time I defended my PhD, and I used to spend a lot of time with him. That's my grandma to the right. She did a master's degree in literature, also at UW-Madison. Both my parents went to UW-Madison and they met there. My daughter went to UW-Madison. So there's a lot of Madison connection in my family. That's my dad on the lower left in the 70s or 80s. He used to teach a very popular course on medicinal plants. So I grew up with that too. Going way back, like where did my people come from? Like they came a long time ago. French, Canadian, Irish. The Cavender was actually Kavanaugh. That's Francis Jakes. He fought in the Civil War. I had relatives that fought in the War of 1812. But then I also had more recent Jewish relatives that came in the 1800s from Eastern Europe. And then my French grandfather there in the middle. So we'll get to France. But my mom grew up bilingual. I have a French name. We all have French names. So we have a French connection too. So that's where I came from. That might have been TMI, but
Click on the little OK button on the screen. Get rid of that one. Get rid of it.
okay.
Perfectly see results.
All right. So getting back to the thing about my dad. So here we are on vacation in the Smoky Mountains. And I thought we were just going for a hike, like we always did everywhere. And so there we are, and my mom's got baby on the back, and you know, click.
Try the arrow keys.
Try the arrow keys. Now I've got the little pen thing. Hang on for technical adjustments. There we go. All right. So this was supposed to be funny, but the idea was when I went back and looked through my childhood history, I realized that everywhere we went, there was a paper about slime molds. So here was like the cellular slime molds of the Southern Appalachians. And here was this trip I remember really well, driving out west from Ohio all the way out to the Badlands, Colorado. My dad and I hiked up this mountain in the Rocky Mountains, and I thought it was just great fun. And then later I find this paper, cellular slime molds of the Rocky Mountains. So that's pretty much how I grew up. And I was mentored, I guess we all were mentored to be biologists from a very young age. I can remember sixth grade staring in a microscope to see mycorrhizae, and somehow I got onto this project of looking at lichens, their diversity and abundance in my hometown. And I started monitoring them for 4 years through high school, and then in upstate New York, where my grandparents lived, and did work on monitoring lichen diversity and abundance in relation to air pollution. So that's kind of where I got my start. I ended up going to the triple, what is it, the American Junior Academy of Sciences to present this work when I was in high school. And I think that was my first time ever in an airplane going out to LA. So that's what I was doing. I was trying to hide this from my friends because it wasn't that cool to be a nerd. So I was also kind of a sports person, and I ran a lot and played basketball and tried not to let people know I was a nerd. Okay, so then Bob said something about my background. It looked a bit like this. So there's my dad, Ohio University, and I really, really wanted to leave Southern Ohio. I just knew the world was a bigger place. And so I begged my parents to, if I could please go somewhere besides Ohio University, where both my sisters went, where we got free tuition. And they said, well, Cornell has a beautiful campus. So I applied to Cornell, early decision, got in, and that's where I went. And I um, I always had this difficulty of wanting to study biology and plants and wanting to save the world. And I couldn't figure out what to do. And so I was always double tracking. And I worked with Sheila Jasanoff, who turns out to be here now. And she was my undergrad thesis advisor. And I was working on the science and policy of acid rain in the United States and West Germany at the time. Sheila introduced me to Bill Clark at the Kennedy School here, and I ended up working in Germany on and off for 4 years while I was doing my master's at the forestry school on learning to manage global environmental risks. And I almost did a PhD with Bill, but then when I was in Germany reading all these Bundestagsdrucksachen, and I was just like, oh my God, I can't do this anymore. I have to study plants. And I wrote Bill a letter. I was like, I can't do the PhD. And he's like, that's cool. There's this guy, Fakhri Bazaz. You can work with him. And so Bill gave me a job for a year, and I applied to work with Fakhri. And that's how I ended up in the biology department at Harvard. And then Missy Holbrook was just hired as a new assistant professor. So I got to work with her and got to go down to Venezuela with Peter and Mary Ashton. I got to be a teaching fellow for E.O. Wilson. I got to know Michael Donahue, and he got me all jazzed. And I met David Ackerley because he was in the Bazaz lab. And all these people were wonderful mentors for me. And I spent a lot of time in Florida during my PhD. I met Karu Kitajima, ended up doing a postdoc with Kath Lovelock, and got connected with Peter Reich. And then we've been longtime colleagues at Minnesota. So on and on. Bob specifically wanted to know about Montpellier. So I will just be totally honest that my reason for spending a fair amount of time doing research in France was more for personal reasons. And I think Serge would be okay with that. I just talked to him yesterday. Serge Ramval was my postdoc advisor. I just had grown up learning French, and I wanted to be able to speak it. And I just, every excuse I had to go to France, and I got to know my relatives in southern France. Anyway, I had done part of my PhD there. And for my postdoc, I proposed to do this project on remote sensing that actually didn't work out, but it was with this guy, Ismail Moya, that I had gotten to know during my PhD. I think 20 years later, they finally got that project to work out, but it just was too big of an ask to get the Paris people to come together with a Montpellier paper to set up the remote sensing. And so I ended up working in a common garden. And I also learned a lot about their drought experiment that Serge Ramval and Richard Joffre had set up. So I spent a fair amount of time working there, measuring photosynthesis in Quercus ilex forests. But what I actually did the work on was the oak common garden that they had set up with these two deciduous, two evergreen oak species, and I was trying to understand their response to freezing stress, given that they have different leaf lifespans. And so here you go, Bob. I don't think I've ever given a talk on this paper, but what I was trying to do is measure them in summer and in winter in terms of their loss of hydraulic conductivity, their loss of ability to transport water after exposure to freezing in these 2 times of year. And it turns out they're a little more resistant to freezing in the winter after they've been exposed than in the summer. But what's really interesting is the evergreen species are more resistant to freezing than the deciduous ones, as you might anticipate, and that their loss of conductivity was related to their vessel diameter. So the ones with larger vessel diameters were more vulnerable to cavitation. And so that's kind of what you're seeing here. And if you look at the chlorophyll fluorescence and how it declines in relation to the loss of conductivity after freezing, there's a relationship there. So the more they lose conductivity, the more they lose their chlorophyll photosynthetic function. And this all seems to be related to leaf lifespan. So if they have longer lived leaves, they're more resistant to freezing, and have less native embolism and smaller diameter vessels. So that's all in 4 species. You can't make too many broad claims about 4 species. But then as an assistant professor, we went on to look at this clade of live oaks that occur in the southeastern US and throughout Central America. And there's 7 species in this monophyletic lineage. Species boundaries are often a bit blurred here. I spent really long time studying the molecular phylogeography of this group, but also their physiology. And in this paper, we found that I can't actually read it very well, but we found that the stem freezing tolerance, when we grew them in 2 different climate treatments, was related to the climate that they were found in. So the ones that were in colder places were more tolerant of freezing, and the ones in warmer places were less tolerant of freezing. And the ones in colder places had greater ability to acclimate to cold than the ones from warmer places. And this was all done in a common garden, so we knew that it had a genetic basis. So MacArthur had this idea that there would be a trade-off. If there was a stress that species were prepared for in the north, that would have a cost. And so they would pay that cost by having a lower growth rate. And what we found here is that there was a trade-off between freezing tolerance and growth rate. And so we went on, and later, I mean, this isn't the only thing we were doing. So this paper only just came out recently, but so we went and studied many species, and found again that the climate that they're from is highly correlated with their vulnerability to freezing and their ability to cold acclimate or express genes that change their cell walls and allowed them to be more tolerant of freezing. Their ability to do that was related to the climate where they come from. And this is for oaks all over the Americas. So that's what we found in this study. And we looked at how long it takes to evolve that. It takes about 9 million years to get halfway there. So they're highly adapted to their climates, but it takes a while to get there. And then we looked to see if there was a trade-off with growth rate. And here we didn't see that. So if you look at the bottom corner there, there's no trade-off with growth rate. So you see growth rate on the y-axis and freezing tolerance on the x. And even though growth rate increases with temperature, the climate of origin, there is no trade-off. And so this was kind of puzzling. And so, in teasing this apart, what we find is it really has to do with leaf habit. And so in the purple there, you see the evergreen species. They have faster growth rates. They also have lower freezing tolerance. But the deciduous ones are the interesting ones. So deciduous ones all lose out on growth rate by being deciduous. So there's this period of time where they can't photosynthesize. So they don't photosynthesize as much in a year, and they don't grow as much as the evergreen species. But that is not coupled to how freezing tolerant they are. So they all have reduced growth rate, but some of them are highly resistant to freezing, and some have lost that freezing tolerance, which is really interesting, which means deciduousness can be beneficial for other things besides getting out of winter. It also helps them get out of the dry season as they move into more tropical realms. So that was an interesting finding in this study, and just keep going. So in addition to looking at freezing, we've spent a lot of time looking at drought. And here's a study where we looked at drought resistance mechanisms in the tropical live oak that occurs down in Costa Rica. And basically, the ones that occur in the wettest area that have the longest leaf lifespan, they're the ones that actually have to make it through the really severe dry season down in Costa Rica. This is in the Guanacaste region. And so they actually have the most plasticity. When you grow them in wet or dry conditions, they can do either thing. They can adjust osmotically so that they're really drought tolerant, or they don't do that. Whereas if they're from the really dry place in Honduras, they just are drought resistant and they can't adjust. So their plasticity has actually evolved. And we spent a long time, I probably spent 10 years working down in Costa Rica, setting up a reciprocal transplant experiment in Honduras and upland and lowland regions in Costa Rica. We did the phylogeographic work, looked at gene flow, set up these common gardens. This was so difficult to do that I can't even describe how many Thanksgivings I spent moving acorns across borders. And I'm not even going to tell you how I got over all those borders, because you have to go through Nicaragua to go from Honduras to Costa Rica. And we were collecting in Belize and Mexico, and it was so many different phytosanitary certificates and import and export permits. It was nuts. So we did that for a good long while. And we published some really good papers and learned about evolution of populations in this system. And then some bad things happened and I left, and that was that. And we continue, though, to look at the evolution of vasculature in oaks in relation to their climates of origin. And this study, this was led by former postdoc Clarissa Fontes, and we went around to different arboreta and we collected woody stem tissue, and then we looked at it under a microscope in various ways. And one thing that's fairly easy to look for, straightforward to look at, are the vessel diameters and the hydraulic vessel diameters, because flow through a vessel is kind of like flow through a pipe, and the hydraulic weighting is the flow rate is proportional to the fourth power of the radius. So you can do a calculation for the hydraulically weighted vessel diameter. But then this other thing is that there are these vasicentric tracheids in oaks that are very cool. And so, yeah, oaks have tracheids, and we often think they're just for mechanical support. But Carlquist had this hypothesis that maybe they're doing something else. Maybe in the oaks, they're actually, and in the various dry forest species, they are playing a transport role. And so what we find is that the more droughted the climate is that species come from, the more vasicentric tracheids they have. And we think that they're like this backup system that goes into operation when the main vessels blow. So oaks have really large vessels, which are very readily cavitated when under tension. So how do they keep going under severe drought? Well, perhaps these vasicentric tracheids, perhaps they help connect up vessels when there's one in between that stops working. So that is an interesting finding based on the vascular system. Then this postdoc, Matthew Caproth, was looking at osmotic regulation, and can't actually see this very well. I will stare closely. So the x-axis for all of these graphs is an index of moisture that was calculated based on how wet it is during the growing season. So towards the right, it's wetter. Towards the left, it's drier. And the leaf osmotic concentration, we grew many oak species in a greenhouse, in a common garden, under low and high water levels. And we measured their osmotic concentration. And if they were from a dry place, they had high osmotic concentration. If they were from a wet place, they had low osmotic concentration. And that allows them to be able to pull water out of the soil at a more negative water potential. We also saw that the stomatal pore area, or if you look at the stomates under a microscope and their size, you just see that there's greater stomatal pore area in species from wetter places. And if you look at the gas exchange, we actually found that the drier site species had higher gas exchange rates. And we think that's because they're adapted to going really fast when they have the water. And then they don't need to go that fast when they always have access to water. So it's actually the drier site species have a greater capacity for gas exchange. And then we can divide those gas exchange values by the stomatal pore area, and you see that there's a very tight relationship. So again, we were finding adaptations in a common garden that showed genetic basis for functional trait variation in relation to climate. Another way we can do that within a species is to plant maternal families within populations from different climatic regimes and plant them out in gardens in different climates. So this is the classic reciprocal transplant experiment. We did this for bur oak Quercus macrocarpa in the Midwest, and at Cedar Creek in Minnesota, in Illinois, in the Morton Arboretum, colleagues there, and in Oklahoma. So 3 climates. We had 20 mothers from each population that we planted out in a quantitative genetics design. And we hypothesized that the local population would have higher fitness in its home garden, and that its fitness would decline in other gardens. But there's also this possibility that, and this work is led by former PhD student Lucy Ray, there's also this possibility that everybody does better in the north because things are getting pretty darn hot in the south. So that was another possibility. And this paper is in review, but I'll just basically tell you that in the southern garden in Oklahoma, the Oklahoma population was the only one that liked it there. Minnesota hated it there, and there was a lot of mortality. In Illinois, everybody loved it. They had perfect soils and it rained all the time. So everybody did great and grew fast in Illinois. If we combine growth and survival, you can calculate a fitness using some interesting models that I'm not going to talk about. But basically, Oklahoma had the highest fitness in Oklahoma in this really red clay soil. In Illinois, Oklahoma was still doing better than the other populations, meaning that it actually liked it farther north, and was doing better than those others. And in Minnesota, Oklahoma lost its advantage. You can sort of explain the growth based on photosynthesis and chlorophyll. And when you look at nitrogen in the leaves, you kind of see, yeah, if they have more nitrogen, there's more nitrogen in the soil. Everybody does better in Illinois. It's just the soil is a better place to be. But this also indicates that actually you could move the Oklahoma population farther north and it would be fine. And it would do well. And it might actually, if you did assisted gene flow, you actually brought propagules and allowed introgression, you might increase the resilience of the bur oaks farther north as genes were exchanged and they start pulling out alleles that help them fare better farther north. So this is some evidence that assisted gene flow would probably work in this species. So we're doing this now for northern red oak, and we have 8 populations, 100 maternal families that we're planning out in 4 common gardens in Maine, at the Harvard Forest in Western New York and in Virginia. This is led by postdoc Stephen Augustine with various collaborators. And there's another grad student doing this with some other species. But there's a genomic component to this, but I'm just telling you that this project is just getting off the ground and we're just getting this started now with the same idea of trying to understand whether we can increase resilience through assisted gene flow and what the genomic basis for adaptive variation might be, what the functional traits are that might allow populations or families to do better in one place than another. Okay, so far, all I've done is talk about oaks. And so this is the part where I feel like I have to justify my existence. Why do I spend so much time talking about oaks? Well, they provide major ecosystem service benefits, and there's 260 species in North America, over 400 species globally. That's a pretty diverse group. Up to 30% of the biomass in North American forests, more diverse than any other woody group in both the US and Mexico. So that's pretty good rationale. I did an economic analysis with some folks from the economic department at the University of Minnesota through the succinct. They let you have these working groups. And so we use the forest inventory analysis data of the US Forest Service and found that oaks have higher climate regulation and air quality regulation value than any other lineage. The pines have more commercial value, more wood product value, but the oaks are really, really important in terms of their sheer biomass for climate regulation and air quality. So we owe a lot to the oaks. And that brings me to the next question of, well, how did the oaks get here? And I'll go through this pretty quickly. And some of you have heard this story before, but I just want to remind you that the biomes we're in now were not always here. And a long time ago, it was tropical. And then as CO2 declined with plate tectonic activity and temperature declined, got cooler and drier, we got dry forest, then we got temperate forest, then we got desert, then we got grasslands, and the Mediterranean system's only like 5 million years. Tundra is only 2 million years. So these systems that we think that we feel so attached to now are very dynamic. And as these biomes emerged, different lineages were radiating. And so at this point, some 40 million years ago, when the temperate zone was expanding in North America, the oaks, who happened to be good at evolving deciduousness and dealing with freezing tolerance, were ready to deal with a cooling climate and with winters, and they radiated. In the two major lineages, they went nuts and they radiated. It was a major adaptive radiation in parallel, sympatrically and in parallel. And I'm just going to say, worked for a long time with my Mexican colleagues, Antonio Gonzalez Rodriguez, and this group of amazing scientists led by Andrew Hipp, and we reconstructed the phylogeny of the oaks of the Americas, and we documented that they actually evolved at high latitude, not out of the tropics, like I was kind of taught in graduate school. Thank you, Michael Donahue. But yes, they did not come out of the tropics. They came out of high latitude, and they radiated. The red and the white oak lineage, the Lobate and the Quercus, section Quercus, radiated sympatrically and in parallel on both sides of the Rocky Mountains. In the green, that's the smaller lineage I talked about before. And this sympatric parallel adaptive radiation explains a lot about the community assembly patterns that I was noticing as a graduate student that I was trying to make sense of. And so what I found as a graduate student was down in Florida, and I found, well, there are these hydrologic and fire gradients, and there are everywhere along those gradients, there are red oaks and white oaks and live oaks everywhere. So they're like radiated across the gradients, and they did so in all the major lineages. And so that was a pattern of phylogenetic overdispersion. And I wrote in that paper at the time, this looks like a signature of adaptive radiation, of parallel adaptive radiation. but we didn't have the data at that time to test it. But then when the Forest Service published all the forest inventory analysis, we could look at all the plot data, and sure enough, the reds and the whites have nearly identical distributions. And this look, if you look at their climatic niches, they're nearly identical except for the Intermountain West. There's Quercus gambellii there. If you look at their functional traits, it shows a signature of convergent evolution. If you look at their climatic niches, it shows a signature of convergent evolution. We looked at a multivariate test of 6 functional traits and did a formal test of convergent evolution. And we find that there's a group of reds and whites that show identical morphology for these 6 functional traits, showing convergent evolution. And so that meant that the oaks were really good at radiating, packing themselves in, and pretty much taking over. And they were probably helping each other, facilitating each other. At least the white oaks were probably facilitating the red oaks. And we wrote this all up in a series of stories, including Scientific American explaining how the oaks took over the Americas. And this has really been one of the coolest stories that I've been part of, trying to understand the role of evolutionary history and diversification in community assembly. And so now that I've explained why evolution is so important for understanding community assembly, I can go to the part where we are really trying to disentangle the importance of community assembly for ecosystem function. And so Bob also asked me to talk about Cedar Creek. So here is the Cedar Creek Ecosystem Science Reserve, where I just spent the last 2 decades. And I led the establishment of this forest and biodiversity experiment with 12 different native species in many different combinations, teasing apart functional and phylogenetic diversity from low to high diversity and different combinations of those. And just as an aside, because I'm allowed to tell stories, the Forest and Biodiversity Fab experiment, we call it, was also Fakhri's initials. We used to call ourselves the Fab Lab. So that's a nod to Fakhri. And by the way, as long as I'm allowed to tell stories, we just got an award named for him and one of his students, Stuart Pickett. It's the Fakhri, Abizaz, and Stuart Pickett Award for Mentorship and Diversity, Inclusion, and Belonging. And we're going to present it to Stuart this summer as he retires the Ecological Society of America. So it took a long time to make that all happen. And it's nice to honor somebody who played such a big role in many of our lives. So anyway, this is the experiment. We just published the methods for this, and my doctoral student, Maria Park, who's here with us now, has been working on this. And because it's such a big experiment, we decided we better use remote sensing. So we've been flying drones and multispectral sensors over this experiment. And the y-axis in all these graphs is fractional cover that we get from point clouds from the LiDAR from the drone. So it basically tells us how much space is filled by the trees. And what you see is that whether you put species richness, functional diversity, phylogenetic diversity, or phylogenetic species variability on the x-axis, on the y-axis, it's predicting fractional cover. So you're getting higher fractional cover the more diversity that you have, and for any of these metrics of diversity. But that's just a pattern. It's not actually telling us about mechanism. And so what Maria is working on for her dissertation, and this paper is about wrapped up, what she has found is that the packing of tree species when they're very diverse has a buffering effect on the microclimate. And so the more packed they are together, the more diverse they are, the more packed they are together, the more that packing buffers the microclimate from extremes, so that the vapor pressure deficit is less variable, temperatures are less variable. And so what you see on the x-axis is that the fractional cover, which was measured from the LiDAR, is negatively correlated with the amplitude of vapor pressure deficit. So more cover, less amplitude. And then the amplitude, the vapor pressure deficit amplitude, is predicting over yielding, meaning how much more biomass do you get as a consequence of different species growing together, as a consequence of diversity. So it's the greater productivity that is yielded than expected based on monocultures. And so she's providing a mechanistic explanation for why diversity begets greater productivity. And I'm explaining this because we're setting up a similar experiment here at Harvard Forest. And so we started on this before we got here. And had I really understood about the glacial till, I might not have taken on such an ambitious experiment, but it's basically been a rock digging mission, even though they hauled a lot of rocks out of it before we got there. We've been digging. Cedar Creek's all sand, so you just it's pretty easy to put a tree in the ground. This has kind of been a nightmare, and it took so long to get all the trees in there. And then we had to go back to Minnesota and pack up everything and come here. And in that time, this was like planted on an old golf course, and God knows what the seed bank was. But when we were too exhausted to get the mulch in the ground, and we had to go pack up all our stuff and get our pets and our plants and all our stuff, and we kind of let the mulch slide, and then this invasive Setaria grass, this European invasive species, just took over. It went crazy. It was like a monster. It just like, I don't have photos to show you, but Setaria took over the whole experiment. And then just this week, after weeks and weeks of pulling this weed out, we finally got the weed out, and now we're finally putting the mulch back in, and we're replanting the dead trees. We'll get this baby in there, but it's been really hard. Okay, so this one is a bit different because we, of course, don't want to do the exact same experiment, but it's a whole different set of species for one thing. And so there's 16 species, 12 different species levels. And a lot of this has a phylogenetic design where close relatives are occurring together or with distant relatives. But there's also species at their northern range limit and species at their southern range limit. And so this gives us an opportunity to test what happens with climate change when some species are getting pushed out of their range and other species are getting pushed into their range. There's also two species in there, the beech and the hemlock, that are just sitting ducks for an invasive pest with the nematode and the woolly adelgid. And so it's also an opportunity to see what happens to community assembly and ecosystem function when you have when you have a terrorist in there. So this experiment's just getting going, and folks in my lab, and a lot of this work is led by Stephen Augustine again, but everybody's out there doing all this. If you want to go out and help plant trees, just let me know. Okay, wait, how much time do I have? All right. I'm going to talk a little bit about spectral biology. I've been working on this, well, ever since France, really, but this has gotten a lot of traction in recent years, and it's really important for herbaria. It's really looking at how reflected light, which is on the y-axis, and wavelengths across the electromagnetic spectrum from 400 to 2500 on the x-axis. This is the visible region here, the near infrared and the shortwave infrared. And we've been finding that it's been known for a long time that it's the spectral signatures are very much related to structure and anatomy and chemistry. But we've been finding there's high fidelity of those spectral signatures to phylogeny. And that finding has really been a game changer for herbaria. And so this was a working group that I led some years ago. And at the time, Dudu Mireles was a postdoc in the lab. Now he's a professor at the University of Maine and just got tenure and career grant, all this. But he led this paper where labs from around the world put their spectral data together, phylogenized it, and found that where you see these colors for these different wavelengths, it indicates phylogenetic signal in that part of the reflectance spectrum. So it shows that where in the spectral signature there is high fidelity, the phylogeny differs for those major lineages, but all across the signature, there is strong phylogenetic signal. And so it gives us a potential means to identify lineages based on their spectral signatures. It's also a means to look at trait and phenotypic evolution back through time. And it's a means to predict all kinds of functional traits. So this is a fresh leaf spectra. Of course, these are all oaks. I apologize for that, but it doesn't matter. They all kind of look the same. These are fresh leaves and pressed leaves. So when you take the water out, they look different. But from those different spectral signatures, you can predict many functional traits and chemical elements. And we use statistical models. They're often machine learning models, but we can also use radiative transfer models, which are physical models to predict certain traits from both fresh and pressed leaves. And so this is really making us all very excited about doing this with herbarium specimens. But not just herbarium specimens, we're also really excited about plants outside and using this method to map plant function over large spatial extents. So here we are at Cedar Creek before we came, like the summer before we came. It was really dry, so we could walk through all the wetlands, and we just went all across Cedar Creek collecting plants, measuring their spectra. And we also, our colleague in Wisconsin, Phil Townsend's group, they flew airplanes overhead. And our plan is to use these methods to map the functional traits of Cedar Creek. We also started our own project on INAT that's getting populated really nicely. It was super fun to just go out and work with all these different plant species. But this is what trait mapping looks like. So this is work from Kyle Kovach and my colleague Phil Townsend, UW-Madison. These are the different neon sites. And this is just plotting functional trait variation for all those sites. So that gets me to this point where I've been leading for the past 5 years, National Science Foundation Biology Integration Institute that we call ASCEND, Advancing Spectral Biology in Changing Environments to Understand Diversity. We just had our 5th annual meeting. And the goal of this is to use spectral information across scales to understand variation within species, across the tree of life, to understand community assembly, ecosystem function, and biosphere function, and to combine spectral information with really careful functional information and process modeling, biological methods using experiments to understand processes across scales using a common data type. And I'll just give you one example. Of course, this is an oak example, but this is oak wilt, which is this most lethal pathogen affecting the oaks in North America. And it's a fungus that spreads below ground through the vascular tissue. The plants form tyloses, especially if it really hits the red oaks. They form tyloses that block the vessels, and then they can't transport water. So they're basically killing themselves. And so we're using spectral information, using mechanistic understanding of how it presents at the leaf level, canopy level, and stand level. Let's see. I'm going to have to skip some stuff or we're going to miss some things. So I'll just show you that we can detect it spectrally at the leaf level. And if we use airplanes and get hyperspectral data, this is across Cedar Creek, we can use that information to identify all the tree species. And then once we do that, we can pull out where the red oaks are. And then when we do that, we can figure out which ones are sick. And so you just look at that to see how this works. If you look at this image, there's the dead guy. You can't see that the ones around it are sick. But if you look at the image below it using spectral reflectance where your eyes can't see, but projecting it in wavelengths that you actually can see now, you see that this spectral information tells you who's sick where you can't see it. And so we just have fancy machine learning methods to do that at high resolution that allows us with high accuracy to find where the sick trees are so that they can be treated. And so we've been working with the National Park Service and the Forest Service to do this. And then this very smart guy, Antonio Guzman, who came with me, he's a research scientist here, figured out how to make this work using satellite data, yeah, by using a temporal series and the disease progression through time. And these are pretty accurate means of detecting oak wilt over really large spatial extents at regional scales to find where sick trees are. And Antonio's also been working with the FIA data from the Forest Service using the hyperspectral satellites that are up there, DESIS and EMIT. And I'm going to go through this really fast, but he's developing ordinations from plot data from the Forest Service. And then he's plotting the loadings from those ordinations on these maps. So basically the coordinates in three-dimensional space from those ordinations on these maps. And then he's using that hyperspectral data to predict the coordinates on those three axes. And using both of those sensors, which are different and have different extents, it's actually the same regions, it turns out, that are very predictive. So that's very cool. It provides that there's consistency in these two very different satellites in the places in the spectrum they're using to predict those axes of tree diversity. And this is just showing you the models for the three different axes. And from that, Antonio is then able to predict where the maples are, beech are, where the pines are, and hey, what's the wood density? What's the nitrogen content? What's the leaf mass per area? And then we just got this really great idea to measure traits using spectra from the herbaria. And these can then go into the ordinations. And then you can use satellite data to predict those ordinations that are based on the functional trait data. And now you're coupling satellite and spectral data from above and what we measure in the herbarium. So this is all justification for why we're going spectral. But first, let me tell you about biodiversity and planetary stewardship. I'm going over time. So what should I do? Okay. I told you this was going to run long. Sorry. Okay. I just have to tell you that I came here very excited to help initiate this biodiversity and planetary stewardship initiative within the Salata Institute because biodiversity and climate need to be managed together, not separately. And so now that we have these technological advances at a time when we're losing so much biodiversity, but there's also been a lot of international effort to address these issues, it's a really good time to have a biodiversity and planetary stewardship initiative at Harvard. And both the HUH and the MCZ are centerpieces of this, and it helps us address our declining diversity, both abroad and locally, take advantage of all these advances in the biological sciences. I've talked about spectral and remote sensing, but we have all these molecular advances too. And this internationally, there's been a ton of work done on biodiversity in parallel to climate, and it's really the right time to be working on this. I thought maybe, I'm not going to say any more about that, but we're just going to keep going. We're just going to carry on. And Harvard has all these incredible resources. It has the herbaria, it has the arboretum, it has the Harvard Forest, which has a neon site. We have all this opportunity to really integrate the biodiversity sciences here. And so that's what we're trying to do. And all these people have been coming to these meetings very excited from across the university, urban biodiversity, global biodiversity monitoring, ocean stewardship, local economies, ethics, culture. So these are just some of the things people are excited about doing. The divinity school is involved. They've got eco-theology, reaching hearts and minds. Okay, so that's all setting the stage for the herbaria being at the center of this biodiversity and planetary stewardship initiative. And here's where I tell you what we're actually going to do and what's new in the herbarium. This won't take very long. I've sort of already explained the things I'm excited about. But what we're trying to do is have a unified biodiversity database. So we're migrating the specified database over to the MCZ base, the Arctos platform. This is going to take a while, but we'll have a unified database that can search everything at once but will also be have user interfaces that are separate. We're very close to getting 2 million specimens digitized, and we're now automating that through Picture-A. And we are holding a workshop in May to bring people together from around the world to test out and agree on protocols for next generation spectral digitization. And we're aiming for 1 million specimens that have already been imaged. So we're going spectral, coupling new spectral information with hundreds of years of accumulated biodiversity knowledge. And we've been running this international working group with Herbarium around the world, sharing how people are doing this. The idea is that if we can agree on our protocols and do things the same way and really hammer things out before we get started, we won't have to backtrack so much, and you can actually aggregate the data. So we're really taking our time to get this right. Also, Harvard Papers in Botany is getting a new look. So Gustavo Romero is retiring very soon, and I did not want this to die. So we are we're allowing botany. As you can kind of tell, I'm not your classical taxonomist. I have a pretty broad view of botany, and so we're going to have a pretty broad view of Harvard Papers in Botany. We're not going to lose the systematics. Now we're going to have a whole editorial board. 3 people are taking care of the plant systematics. We're maintaining multiple languages, including 2 people are native Spanish speakers. But now we'll have quantitative genetics, ecology, fungal biology, plant ecology and biogeography. And we'll even have some remote sensing. So this will be much broader. We're going to have the papers be largely invited, but if you want to be invited, all you have to do is write a proposal to one of the subject matter editors. Another thing is, we'll get ready. I mean, it's a bit of a ways off, but Jenny's sitting here and can tell us more. But there's going to be this really cool exhibit on plant families where Wendy Hollander's beautiful paintings will be paired up with glass flowers from the same families, and public can learn about phylogenies and plant families. And then, very last, the herbarium is getting a new look, I think. Like, I don't know if the capital projects are frozen or if they're not frozen. The dean said maybe on Monday that they're not frozen anymore. So maybe this is happening. Anyway, we've been planning for months and months a redesign of the front part of the herbarium to make it more accessible and make the space more efficient, more usable, better for collaboration. And so there'll be a beautiful meeting room. We'll be able to have events, dinners in there, and already they've put in collaborative space. So it will look quite different once this actually happens. I don't know how soon this will happen though, given all the issues going on. So this is just a list of all the things I basically already told you. This is what we're trying to do right now. And I keep getting pushed back to my little spot here. But this is what we're doing. We've been working really, really hard on all of these things. And it's, with 5 million specimens, nothing moves fast. But things are happening, and we're really trying to take care of this collection, put it in the center of biodiversity and planetary stewardship, have new exciting things happen in the spirit of the extended specimen that was really promoted by the national academies, and we're doing it. So that's what I have to tell you. Thank you.
Since I'm talking now, I'm going to take a first question. Which oak species are you going to look at here in Massachusetts?
Oh, you mean besides the Quercus rubra that we're doing the experiment with?
Yeah.
Well, let's see.
Repeat the question.
Which oak species are we going to focus on here in Massachusetts? Well, okay, I'll just have to say that Quercus coccinea is really confusing here,
It's what?
really confusing. So I never met such confusing Quercus coccinea before, but it seems to be difficult to differentiate from rubra sometimes. Does this happen to you or is it just me? Bare oak, that looks cool. What do you think I should be focusing on? What's the coolest oak here?
Well, I mean, I think the two scrub oak species would be good. Fair oak and white oak form of that. It looks like Alice, whatever, oh, that's Hills oak.
Palustris? Is it,
Yeah, Brinoides, thank you, yeah.
okay.
Relative to
Yeah, so I
dry,
think
sandy terrain.
we saw that out on Cape Cod. Yeah.
There's a lot of them.
Yeah, so can we go on a field trip and look for them?
We have Palustris coming in from the south. We got Muhlenbergia in the Berkshires. Lots of good species. to look
So
at.
I would be all in on an oak field trip. By the way, the International Oak Society is coming here in 2030. It's ways off because they have to move around continents, but we will be hosting the International Oak Society in 2030. Yeah, please. Well, so the question is, spectral information seems to entail a lot of computation, and that is true. So it's high dimensional information, meaning for a single measurement, you get many, many values. You might have 150 wavelengths that you're simultaneously taking a measurement for. That's a lot of information. And then if you have many, many measurements and many, many species, and then you've got imagery with pixels, and each pixel has a signature in it. And quickly you're having terabytes of data that you're having to process. So that is all true. That is one of the reasons why one of the components of this biodiversity and planetary stewardship is actually having a biodiversity data center and processing center, because this is pretty hard for any individual to do on their own. But if there's a center that has workflows and people that are running those workflows, it makes it easier for individuals to take advantage of those, where there are people dedicated with the expertise that know how to do them, and there's the capacity to do that computation. So the answer is yes, but there's also the capacity for dealing with that already, and we're trying to make it easier and more accessible for people. Yeah, hi.
Question?
Yeah,
So one of the great promises of iNaturalist was that students could just take their camera or their phone and take a photograph of a plant and identify it. I mean, is hyperspectral imaging ever going to get to that point where we just take an image of a plant with this technique and identify the plant? And could you contrast hyperspectral imaging with the potential for iNaturalist to do it?
oh, okay. So actually a graduate student that I co-advise over in the UK with David Coombs just did a study comparing the use of spectra versus imaging, RGB imaging, running it through machine learning algorithms to see which one predicts taxa better. Turns out the images work better. And that's particularly because if you've got a specimen with reproductive structures on it, that's kind of the key to correct identification. Whereas the spectra are really just coming from the leaf tissue, and they're agnostic to the reproductive structures. I mean, it works pretty well, but not as well as the images with reproductive structures. So what that means is it's probably helpful to get at specimens that don't have reproductive structures, or at least to narrow it down to a family, or to get it in the ballpark, because it does work pretty well. It's just the accuracy is not as high as the image algorithms. So that's what we know right now. Could improve in the future, but I think the difference is really just that when you're taking leaf tissue, you're not going to be as diagnostic as when you're taking multiple parts of the plant and the reproductive structures. So how much dry tissue do you need to harvest from specimens? Okay. So actually, when we have this meeting, and the first part of it will be open to the public and live stream. That'll be on May 2nd. So if you want to see what people are doing, you can tune in then. In theory, all these measurements are non-destructive, and you basically have to have a non-reflective background that you slip under a leaf. It has to, if it's glued down to the paper, it's not going to work. And in Harvard, we're not even going to bother measuring them if you can't slip black underneath the leaf. But you don't have to remove any tissue. What you need to remove tissue for is if you want to validate the trait prediction models, but you don't have to use herbarium specimens for that. So you have to actually grind up the tissue and measure the concentrations of chemical concentrations some other way, and measure the spectra on that tissue. And then you develop a model for predicting the trait or the chemical concentration from the spectrum, having measured it another way. And then you can apply that model to a herbarium specimen. So in theory, it's possible to do this non-destructively on specimens. How much tissue you need to build the models is just depends on how much tissue you need to measure those traits the way you normally would. So if you need a little bit of tissue to measure nitrogen concentration, that's all you need. If you need a lot of tissue to get at carbon fractions, lignin and cellulose and hemicellulose, then you need a lot of tissue. So it's just like you would normally do. To build those models, you need to get enough data of the actual traits for, so hope that made sense.
We're working on forest trees. Are other people working on grassland?
Yeah,
Biomes that are-
yes.
I assume it's translatable to our.
Yes, so I showed you the trait maps. They're just sort of like beautiful, colorful pictures. Those are for all the neon domains, which are across all the major biomes. In Cedar Creek, Cedar Creek has 3 biomes, including the prairie grassland. So yeah, this works for any kind of vegetation. You can do it for soils too. I mean, it's not just for plants. There's a whole community of people working on soils. So we just happen to enjoy working on trees, but it's not specific to particular group of or habit. Yeah, go ahead.
One more. Does hyperspectral imaging tell you something that you couldn't do using other methods, or just that the advantage is that you can do it faster or non-destructively? Is that the advantage?
Okay, so first, let me just say that there's a difference between hyperspectral. I know you know this, but I know that some people are probably confused about this. But when we talk about an image, we talk about many, many pixels, and each pixel has a spectrum in it. So a hyperspectral image is a very large bit of data because it's got all the pixels plus all the spectra. What we're talking about with the herbarium specimens is really just point measurements. It's just the single spectrum that we get. We just take a measurement, we get one spectrum. It's not an image. So we're not talking about imaging the herbarium specimens. We're just talking about taking point measurements. When you're asking about hyperspectral imagery, like we would get with a drone or an airplane or a satellite, I mean, one thing to keep in mind is the pixel size. If your pixel size is 10 meters, you're going to get a single spectrum for the whole 10 meters. You might be able to identify a tree. You're not going to be able to identify a grass with that. If you use a drone, your pixels might be 0.5 centimeter, and then maybe you're going to be able to get the grass. So the grain size of the pixels, the resolution, spatial resolution of your imagery is going to determine what kind of information you're going to be able to determine from the imagery. So if you're going to try to identify species, you need pretty high spatial resolution. If you want to predict traits and you're happy with a weighted mean, then a large pixel size is fine. And in fact, what I showed you from Antonio's work using the forest inventory data, those are satellites, the it's European and a US satellite, they all have, their highest resolution they're ever going to have is 30 meters. So you're never going to pick out an individual tree. That's why it was kind of a clever way that he chose to do that using an ordination where you're predicting the coordinates in an ordination. So you're saying, this is a community that falls here in the sort of oaky part of the ordination, and here's the pine part of the ordination, and we can predict where in the ordination it is, but we're not picking out individual tree crowns like we were with the airborne imagery, where you could actually get pixels over the canopy and you can use that to identify. Those are pure pixels of one species. So those are different ways of solving this problem of monitoring biodiversity, basically. Did that make sense? Okay, yeah, all right. Okay. Well, we just want to thank you so much again, Dr. Cavender-Bares, and we want to thank you all in our live audience and our Zoom audience for attending and being with us tonight. We're going to adjourn our meeting and move on to refreshments here at Harvard. So thank you all for coming.